It's a great honor to welcome our keynote speaker, Professor Dr. Fernando Stefani. Dr. Stefani is a principal researcher at the National Scientific and Technical Research Council, CONICET, a full professor of experimental physics in the physics department at Exactas, and the director of the Center for Bioscience Research at CONICET. Internationally recognized for his pioneering work in nanophotonics and super-resolution microscopy, Dr. Stefani earned his doctorate with highest honors in Germany and completed postdoctoral research at leading European institutions. Upon returning to Argentina, he established a world-class laboratory at the University of Buenos Aires and CONICET. Dr. Stefani's research has advanced fluorescence imaging and nanotechnology, resulting in worldwide recognition for his breakthrough visualization methods. His leadership has transformed his laboratory into a regional hub of innovation, inspiring and supporting the careers of over 30 young scientists, many of whom now work in academia and industry worldwide. With over 90 publications in leading journals such as Nature, Science, and ACS Nano, and a series of prestigious awards, including the Otto Hahn Medal, the Georg Foster Research Award from the Alexander von Humboldt Foundation, the Hussey Prize in Mathematics, Physics, Astronomy, and Computation, and the Konex Prize. Dr. Stefani ranks among the most influential scientists in Argentina over the past decade. Beyond his research, Dr. Stefani is a dedicated mentor, a strategic advisor to scientific institutions, and a passionate advocate for advancing science in Latin America. Please join me in welcoming Dr. Stefani, whose vision and achievements continue to shape the future of nanoscience and bioimaging.
Okay, wow, Leah, that's a bit too much. Thank you very much. Good morning, everybody. So, I'm gonna use the few minutes I have this morning to talk to you about technology applied for bioimaging, and particularly to bringing fluorescence microscopy down to molecular level as much as possible. So before I start, just a few words about where we work. It's at the Center for Bionanociencias Research. It's not far from here in a new complex that's called the Polo Científico Tecnológico in Palermo. That's a nice building. We are somewhere, maybe I can use the laser pointer. So we are somewhere in the first and second floor here in this building. It's a relatively small research institute, but we tackle interdisciplinary research among chemists, biologists, and physicists. Here you see a few snapshots of my group over the years. The work I will show you today has involved Francisco Balzarotti, Luciano Mazzullo, Martín Bordenave, Federico Barabás, Cecilia Sasa, Lucia López, Mariano Varela, let me see, Florencia Adorna, Florencia Choque here, Santiago Sosa, and Gonzalo Escalante. I hope I'm not forgetting anyone. So, we are a small group, but we are lucky to have a strong network of collaborators, both in Argentina and around the world. And some of the experiments I will show you today were done with the great help of Stefan Hell and Tom Jovin in Göttingen, Philipp Tinefeld from LMU Munich, Alfredo Cáceres from Córdoba, Sabrina Simoncelli from London, Guillermo Acuña from Fribourg, Switzerland, Oscar Campetela and Juan Mucci from UNSAM, Buenos Aires, and Fernando Goldbaum from Fundación Instituto Leluar in Buenos Aires, and Marcelo Martí and Matthias Blaustein in University of Buenos Aires. So let's get started. But just before, I'm quite sorry that the first paper I will talk about is this one that just appeared in Nature a few months ago. And I will not go into details and be reiterative about the difficulties we are going through in Argentina, but just believe me, it's really serious. Nevertheless, we continue working and we have exciting projects, and I will show you some of them. So let's get started. To this community is, I guess, totally clear that fluorescence microscopy is one of the preferred tools to visualize soft matter and particularly biological matter. That's because fluorescence microscopy gathers a set of features that are quite unique. So it gives us like a very high sensitivity and specificity. We can even detect single molecules. It's non-invasive, so we use basically visible light, and it can work at physiological conditions. That means room temperature, liquid environments. And it also allows us to do 3D imaging. So due to this, fluorescence microscopy is really, really a powerful bioimaging tool. But it has a limitation, as you all know. It's resolution, limited by the wavelength of light. So what you see here is a baby neuron imaged with fluorescence microscopy, and it's, as you can see, it's great to see the morphology of the cell, to see how proteins are organized in different domains of the cell. But if we want to look at this with more detail, like zooming in on this growth cone, we see that the image gets blurred. And that's not because we did anything wrong, that's just because we are reaching the resolution limit of light microscopy, which is about a couple of 100 nanometers. So here is just a few slides I'm going to use to explain how bad it is. So imagine a molecule. That's one of the typical fluorophore. It has a size of about 1 nanometer. If we take an image of a molecule with our fluorescence microscope, we get a signal that looks like this. It's about 200 nanometers. So you see, we start with an object 1 nanometer in size, and we end up with a signal that is 2 nanometers in diameter. So in area it's 40,000, and in 3D the zeros continue to increase. So it's really a lot what we are missing. I typically use this analogy with a football stadium. So if you watch a football match, a human being has around 1 meter in size, order of magnitude, and the football pitch has about 100 meters. So this is a size mismatch of 1 in 100. In fluorescence microscopy, you are a factor of 2 worse in just one dimension. So if you would watch a football match with a 100-meter resolution, this is what you would see. Almost nothing. If you are lucky, you could tell there's a football stadium there. But there's no way to tell what the players are doing. There's no way to tell if they are playing together, playing football or dancing or fighting each other. And luckily, a few decades ago, this was partly solved with super resolution microscopy. And this allows us really to see with much higher detail biological structures. Here you see an example of microtubules in a neuron. On the outside of the circle, it's confocal microscopy, and inside, one super resolution technique, STED in this case. And you can see that microtubule bundles that were not possible to visualize before, now you can discern them. You can really see them. Here you see another very nice example from the group of Stefan Hell. Here, 2 proteins of the nuclear pore complex were labeled, and that's how they look in confocal microscopy. Now, if you apply super resolution, this image transforms into this. So the difference is really impressive. That's before and after. So super resolution microscopy is a true discovery tool. There's no doubt about it anymore. And another wonderful example is the membrane-associated periodic skeleton of neurons that was discovered in the lab of Xiaowei Chuang. And since then, it was confirmed in all types of neurons investigated so far. So you see on the left the original image, and on the right one image from our lab. So you see, if you would image this protein with a conventional microscope, it looks like the protein is perfectly distributed, uniformly all over the cell. But when you apply super resolution, you discover that that's not the case. The protein is organized very regularly with a period below the diffraction limit, and that's the reason why it looks so uniform. So this is really a discovery, and it actually is such an impactful discovery because this structure has been found in all types of neurons, in all organisms studied so far. So basically the whole neurobiology needs to be revisited in view of this, because everything we have thought so far about how neurons work, we're not taking into account this structure that it's everywhere. So you need to rethink everything. These kind of discoveries are being made nowadays with super resolution microscopy in different fields. And that's why the Nobel Committee awarded 3 of their pioneers with the Nobel Prize in Chemistry in 2014. Okay, so super resolution microscopy is really wonderful, and you have a wide library of protocols that you apply in scanning microscopes, like confocal to photon microscopes, and in wide field microscopes, and they all work very well. Before I continue, just a word of caution, because there are around there algorithms that promise you things like one-click super resolution, and you have to be very careful. That's not possible. You cannot create information, or you will create artifacts. And here you see some examples. Here you see a confocal image. That's a state image where you reveal the periodic structure of spectrin. But of course, from this uniform intensity, you cannot reveal this just by one click. You would get very strange things. So just be careful. And now, to continue, and let's go to the focus of this talk. I promise we will try to bring super resolution down to the molecular level. So I showed you, that super resolution really reveals subdiffraction features, but it has a limit. And that limit is not fundamental, but it's practical. So if you see this beautiful image of the MPS, you will find that the resolution there is laterally of a few tens of nanometers, and in the axial direction is a bit worse, 2-3 times worse. If you look at these microtubules, of course you can see them much better with super resolution, but if we would like to zoom in and look at the supramolecular structure of microtubules, the image gets blurred again due to this resolution limit. It is known from in vitro experiments and X-ray crystallography and that microtubules are formed of this quasi-helicoidal structure of alpha and beta tubulin. But if we want to see that structure in cells, we cannot. So the resolution is not high enough. That has motivated several groups around the world to try to push super resolution to reach a limit beyond 10 nanometers. And that's not because it's a round number. That's because 10 nanometers is about the size of structural proteins. So if we can surpass that limit, then we will be able to start seeing how proteins assemble in supramolecular structures to form these higher hierarchy structures that give rise to subcellular structures, cells, tissue, organs, and so on. And the limit to that actually is not fundamental, it's just practical, it's technological. The real limit is that fluorophores, fluorescent molecules, cannot give you infinite photons. So molecules, when you try to extract fluorescence photons from them, you excite them, they return to the ground state and give you a fluorescence photon, and they do that millions of times. But eventually, they use that extra energy to undergo irreversible photochemistry, and the experiment is over. So you have a limited number of photons you can get from a single molecule. And that's the real practical limit. So there's basically 2 ways to solve this. You either get more photons, or you either get more information from the few photons that you have. And both avenues have been explored and have been successful, actually, in achieving higher resolutions. In getting more photons, you could think of that as the chemist approach, right? And one very successful approach has been DNA paint. I don't know if we'll have time to discuss that in detail, but there have been advances also in stabilizing buffers, self-healing dyes, and other chemical approaches. The second avenue is getting more information from the few photons that you have, that's probably the most physicist or engineering approach. That consists of developing new methods that can give you, with the few photons you have, a better resolution. And that's where our group has worked over the last 10 years approximately, and we have been successful in a few methods that can give you a resolution below 10 nanometers. I will now show you very briefly some of those methods and illustrate what you can achieve with them. So basically I will discuss three approaches very quickly. The first one is called simpler. And so this acronym was made on purpose, of course, because it's a really simple method which consists on calibrating a TIRF microscope. Just doing that, you can achieve a very high resolution. A TIRF microscope is a fluorescence microscope that uses total internal reflection to excite the molecules. So the light doesn't propagate into the sample, but you illuminate it with an evanescent field that is maximum at the interface of your sample and decays exponentially towards the interior of the sample. It looks something like that, the intensity of the light. So that would already give you an effect that would make molecules brighter when they are closer to the interface. But there's something else. Molecules also emit differently when they are closer to an interface. A molecule that is closer to a dielectric interface emits more photons into the higher refractive index medium, in our case the glass substrate and our objective. So we have a second phenomenon that makes molecules brighter when they are closer to the interface. So if you combine the two properly, you can calibrate your microscope. I will not get into details, but trust me, it's quite simple. And you need just to measure the intensity of a molecule exactly at the interface, which is easy. You just throw antibodies or fluorophores and you measure the average intensity of them. And now you can extract the axial position of a molecule just measuring intensity. And it works very well. Here you see an example of microtubules observed by Storm, and this is a 2D image. Now applying this method you can convert it into a 3D, and you can you clearly see the cross-section of microtubules. So here you are seeing the lateral cross-section of microtubules, which have a size of about 25 nanometers. When you label them, they are a bit larger, but these are very small structures that you can resolve on an optical microscope. You can also see microtubule bundles that otherwise would look like a single microtubule. And now, when we looked at this, then we had a resolution of about 8 nanometers. So we thought, okay, we can see the quasi-helical structure of microtubules. Let's take a look. So we were very careful to look for straight microtubules in the XY plane, in the XZ plane, and try to look at the microtubule internal structure, but we couldn't see it. We couldn't see it. And that's because the labeling efficiency, the labeling density is not high enough. But nevertheless, since we have the resolution, we can apply very simple statistical methods like quantifying the distance to the first five neighbors, and then we see that those distances peak periodically at about 8 nanometers. So we can reveal that there is an underlying structure with a periodicity of 8 nanometers in this case, confirming what people have seen in vitro and electron microscopy and X-ray crystallography. So you see, this is one first example, and I will give you another one later, that reaching this level of resolution allows fluorescence microscopy to enter the realm of structural biology. And that's something really interesting that I think will develop more and more in following years, because we can perform experiments inside cells at room temperature in liquid environment, and this allows a new dialogue between structural measurements and simulations. Well, in this case, this method is just calibration and software, so if you are interested in applying it, you can download the software and also contact us if you need help, of course. All right, another way of achieving this kind of resolution or measuring molecular interactions with fluorescence is a combination of STED and FRET. So probably all of you know FRET, so I will not go into details. But I will show you this example. I will use again this wonderful example of the MPS of neurons. So the working model for this is actually, it looks like that. We think this structure is made of actin rings that are separated very regularly by spectrin tetramers. that gives this spacing of 190 nanometers. And we believe that adenosine, another protein, is actually performing the link between spectrin and actin in those rings in this way. So this is the working model. So how can we test it? We would like to know, for example, an important piece of information would be to know if actin and adenosine are actually interacting in these rings. So that very simple question could be resolved with FRET, you would say. Yes and no, because the problem is that if you would perform a FRET experiment in a conventional microscope, then you would be measuring something like that. So even if you detect FRET, there's no way you could tell if adenosine and actin are interacting inside the MPS or outside, because you're averaging a larger area. So our idea is to combine FRET, but to measure it inside a subdiffraction area limited by STED. And this already gives you a much better information, because now you can tell inside the small circle if molecules are interacting. So I don't have time to give you all the details how this works, but basically it's a two-color STED measurement. We tested this using DNA origami. It's a wonderful technology to test super resolution methods, and we could confirm that we could see super resolve interactions through STED. And you need to perform some controls, but here you see a result. Here's a STED measurement, a FRET measurement, diffraction limited, and you would see that adenosine and actin are interacting somewhere in the cell. But it's very hard to tell anything from this. But when you combine it with STED, then you can find that the interactions present the same periodicity as the MPS. And then you can confirm that that's an active site for interaction between these two proteins. All right, and now the last minutes I have, I will use them to show you a family of techniques that we called single molecule localization with sequential structure illumination. And this all started trying to improve the localization precision of a single molecule using fewer photons. So the typical way to localize a molecule is using a camera. So you illuminate your sample with a uniform intensity, and then you use a structure detector, like a CCD camera, to collect photons of a single molecule, make an image, and then you would pinpoint its position by fitting some function, right? And this works, of course, but the localization error follows this formula. It basically, of course, it improves with the square root of the number of photons you collect, but here in the numerator, you have the PSF, you have the resolution limit. This is about 200, 300 nanometers. So you're fighting these 300 nanometers with photons. That's a problem I described at the beginning. So we tried to find another way of doing this and getting rid of this large number in the numerator. And one way we found was localizing the molecules using a light minima. The first implementation of this was a minflux, and the idea is to kind of triangulate the position of a molecule using a minimum of light. Since we used this beam with a circular symmetry, we needed to use 4 positions, not 3, to localize a molecule in 2D. And this works very well. Here you see an example. Here below, you see many localizations of a single molecule using just 50 photons. And you see the cloud of localizations disperse over 10 nanometers only. If you use 500 photons, then your localization error becomes already 3 nanometers. And if you can use a few thousand photons, you already get into the 1 nanometer regime and below. This works very nicely. But the key point here is that, of course, the error still scales with the number of photons, just like before. But now if you look in the numerator, we no longer have the diffraction limit. We have L, which is an experimental parameter. L is actually the distance between the minima. And we can make that smaller and smaller if we need higher localization precisions. When this was first published, it generated a big impact, especially because of this possibility of zooming in. Here you see the localization error as a function of the number of photons. They all scale with the square root of n, of course. But what you see here, these 3 curves, are just localizations made with the decreasing size of the field of view, L. So with the same number of photons, we could get something like 5 nanometers using an L of 150 nanometers, but if that's not enough, we can zoom in, and then our localization error becomes 3 nanometers, and we could zoom in further to 50 nanometers, and then we get into 1 nanometer. So it's a very powerful method to localize more and more precisely at the expense of measuring in a smaller area. And this can be applied for imaging, like you see here for this, where we tested it with a DNA origami. You see the scale bar here is 10 nanometers. So we can clearly resolve things below 10 nanometers using an optical microscope. But it's also very useful for tracking, because since you have to localize things with fewer photons, now you can track faster. In this example that you see here, we were tracking ribosomal proteins with a modest spatial resolution of 50 nanometers, but to achieve that, we only needed 9 photons. So we could track very fast. And actually, for tracking, it's better to have higher temporal resolution than spatial resolution. That's why we chose that combination. And that allowed us to get a diffusion coefficient at video rate. So this is D as a function of time with a 30 millisecond resolution. So instead of having one image every few tenths of milliseconds, we had tens of images. And we could see how proteins diffuse, stop, diffuse again, and we know, of course, where it did it. So in terms of instrumentation, MinFlux, it's quite complex. The first implementation was using electro-optic deflectors and electro-optic modulators. The setup looks simple, but trust me, it's quite complicated, and could achieve this measurement at 8 kilohertz. So that motivated us to make a simpler or at least easier implementation using pulsed lasers, and we did that a few years back, and that allowed us to measure this at megahertz. So now we can make these measurements like a machine gun, and then eventually we detect the photon, and we can know, using time-correlated single photon counting, we can know from which donut came that photon and reconstruct the position, and at the same time we get the lifetime of that molecule. And that opens up also new interesting possibilities. Here you see some wonderful examples that we did with a group of Professor Tinefeld in Munich, where 2 molecules, spectrally identical, could be localized simultaneously, just multiplexing with lifetime. So I think this is the first time where 2 molecules could be super resolved without blinking. And that gave rise to several different possible applications that I don't have time to go through right now. We could even, using the lifetime and position information, getting to the Fret regime of these 2 molecules and still find their position below 10 nanometers. So, I think I have just a few minutes. So, I want to stress that the key point of these methods is basically on using a minima of light. So, this idea of zooming in further and further to localize a molecule more and more precisely it comes just from using a minima. So what you see here is a parabolic minimum, right? If you zoom in, so if we expand this area and we rescale it, that minima looks the same. And if we do it again, that minima looks the same. And we can continue doing this forever. It's a scale invariant problem. So that's the key. And then I showed you that the first time we used this triangulation thing to localize a molecule inside that minima, but actually, since the key is the minima, it doesn't really matter how we probe that minimum. So we can probe it with these 4 points, but we can also probe it with an orbit, or we can probe it with a much more common raster scanning, like in any confocal microscope. And that works just as well. A few years back, we made a theoretical paper where we evaluated all kinds of methods based on this concept using a light maxima and a light minima. And all methods using a light minima allow you to zoom in indefinitely and achieving these types of localization precisions. So from all the methods that we evaluated, of course this one, RASTMIN, came to our attention because it can be implemented on any scanning microscope, which are widely available around the world, like confocal microscopes, confocal or two-photon microscopes. So all you need is a scanning microscope. You need to add some stabilization, and you need to modulate the phase of your excitation beam in order to have a focus with a central minimum, not a central maximum. That's all you need. And once you do that, all you have to do is to scan a small area. So here what you see are single molecules on a scanning microscope where you changed your focus to have a central minimum instead of a maximum. Then all molecules look like donuts, of course. Then you pick one and you make a small scan of about 100 nanometers. And you make that scan, and of course the molecules looks like that. It looks like a minimum. And you can localize it using 200 photons or 900 photons or a few thousand photons. The minimum, of course, looks better and better. You would say this is almost the same as localizing with the maximum, but it is not, because the key, which is the difference here, you can zoom in more and more, and the minimum always looks the same. So the error will scale with the size of your scan for any given number of photons. So here you see and some experimental data to confirm this. Here we localized molecules many times using different number of photons. The red curve is the theoretical limit, and the gray shade is actual practical limit, which considers the theoretical limit and how good we can stabilize our microscope, which in our case has an error of about 1 nanometer also. And then you see the experimental points, which fit quite well. So let's see if we can use this for imaging. All you have to do, as in any other case, when you have a localization technique, you need to let molecules blink now and localize them one by one. And again, we use DNA origami to test this. In this case, an origami with 6 fluorophores. So the origami, when they look like that, again, like donuts, then we pick one and we scan continuously a small area. And we let molecules blink, and you see the experiment looks like this. So you're scanning a small area, and eventually a molecule switches on, and every time it does, you can localize its position by fitting a minimum. And if you do that many times, then you end up with something like this. You clearly reveal the position of the fluorophores. And this is just to tell you this is done in a scanning microscope just with very few modifications. Okay, they are telling me that I have to hurry up, so I will speed up. Here you just to compare the levels of resolution that you can achieve. And diffraction limited image first, then super resolution of the first generation, as we call them, and then Rastemin or MinFlux or one of these super highly precise methods. And you can really reach about 1 nanometer and below resolutions. So to finish, I want to stress this new direction that I think it will become more and more interesting the next years, which is addressing structural biology using fluorescence. And here you see 3 papers from other groups, particularly for the group of Stefan Hell and Ralph Jungmann, where this is starting to happen because the resolution is high enough to visualize structural details of biomolecules at room temperature, in liquid environment, and sometimes even in live cells. And to finish, just an example from our lab where we addressed a new dimension of this. So positions are very precise, I showed you. Another interesting dimension of these measurements are orientations. It's very important to determine orientation of molecules. And here we show one way of doing it where we detected the orientation of a single fluorophore inside a protein cage. And this is a very small protein. It has about 8 nanometers and 6 nanometers has the size of a typical structural protein, and we could put the fluorophore inside and detect its orientation inside that cage. Which to our, I cannot explain the measurement techniques, but it's not so complicated. What I really want to stress is that we could determine the position of that molecule inside that protein cage, which is quite small. We could, this was done at room temperature and in liquid, so we could see actual dynamic behavior. We see the molecule jump between orientations, and we found out that that molecule takes only 2 preferential orientations, and we could detect them. And when we compared that to molecular dynamics, we found a quite good agreement that is really surprising and encouraging, because now we have measurements that can be compared in a much better in a much fair terrain with molecular dynamic simulations, because typically we have X-ray crystallography or cryo-EM data obtained in superstatic conditions, frozen conditions literally, and we compare them to molecular dynamic simulations, which is the tool we have to explore conformations. Now we have a tool that works at room temperature, liquid environment, so we can compare this to simulations much more fairly. So I think a new dialogue between experiments and simulations will be established, which of course it requires that each experiment needs to be designed to probe a particular feature of that structure. It doesn't have such a throughput as X-ray crystallography, but for particular details, I think it will become a very interesting and complementary technique. All right, with that I finish, and another advertisement of our workshop that was last week, and I hope we can hold it next year, so stay tuned for those of you who are interested in super resolution microscopy. And with that, I thank my group, and I'm happy to take any questions you might have.
Thank you, Fernando, for a wonderful presentation. So I'm glad to see that you're moving very fast to the structural biology field. And as you mentioned, I think it's a very powerful combination of techniques, especially if you combine with things that, for instance, Jose Maria Carazo does with deformations and mathematical models to predict protein dynamics. And one thing that I was very impressed that you can localize very well the protein in the Z orientation, right, Z direction. And this is a big problem when we do correlative work with cryo-EM. So how do you see you applying this type of technology to freeze down, so to localize a molecule, then freeze down the cells and go to the cryo-EM and localize them precisely?
Oh, you mean on the same sample to make like a correlative microscopy?
Yeah, correlative.
Well, I don't know, honestly. In my view, these methods will complement cryo-EM data. So cryo-EM and X-ray crystallography still give you a very high throughput and higher precision, right, down to a few angstroms. I think this method will be helpful to confirm structures. And like if, okay, you have some structure that you found in X-ray or cryo-EM, say, okay, I want to see if that structure really is present in a cell under physiological conditions, then you could design an experiment to probe key features of that structure and confirm its existence. I think that's the main application of this, for the moment at least. Yeah.
Yeah, I agree that it's really powerful, this complementation, and on the side of cryo-EM, there's the advantage that you don't need to design a probe, right?
Exactly.
The structure is there.
Exactly, the structure is there, and so the throughput is higher still. Here you need to design a probe and a labeling strategy, particularly to test that the existence of that structure. Yeah.
Thank you. So a very, very nice question. And talk in general, and certainly we are looking for an integrative approach that mix different techniques and give a better understanding of what is going on. In terms of cryo-EM, really something that we are missing normally is the time domain. You know that there are time cores. I mean, it's difficult and all that. And when we think in time, we are thinking to use molecular dynamics, but it has to be fully atomistic molecular dynamics, because otherwise you don't have the time information. So the systems have to be small. So we are in the catch-22 there,
Right.
So
No, that's a very good point. So the system has to be small, and also the time scales probed are small. So when you do a molecular dynamic simulation, it runs for very short times, usually much shorter than any time resolution that we have in the experiment. That's actually, it was quite surprising for us, because that the agreement was so good. Oh, sorry, my presentation. Okay, because our measurement resolution was of sub-second, right? But it's milliseconds. And the molecular dynamic simulations runs for a few nanoseconds, right? So there's a still in the time domain. But it seems that the conformations that we found to be stable on the few nanoseconds also in the seconds. So those are really preferential orientations. But that is something that now we can probe, you know, because now we can see, okay, what we see in MD, how real is that? But it's just starting, so this is the first time we do it. Now we would like to change. This was I didn't give you the details, but this was a fluorophore with 2 sulfonate groups that we expected to form hydrogen bonds inside the cavity. So something we could clearly do now is to change those functional groups, or leave only one sulfonate group, for example, and see if one of the orientations is broken or weakened. So I think now we need to start collecting data and building a whole formalism and way of reasoning in these experiments.
absolutely. Thank you very much.